Good day everyone. Today we'll be discussing about the method for body fluid identification related to sexual crime based on the journal that we had choose. Now let's talk about the purpose and method used in identifying this fluid. So biological fluid identification plays a vital role in linking suspect to crime, especially in sexual assault cases. It helps provide key evidence such as DNA and confirm whether the suspect genetic material is present in bodily fluid found at the crime scene or on the victim. So the main goal of identifying body fluid is to establish a link between the suspect and the crime scene. Techniques like DNA analysis, mRNA profiling and DNA methylation are being used to identify body fluids such as saliva, semen and vaginal fluids. So this method helps forensic scientists confirm the biological origin of evidence found at the crime scene. So for the relevance for DNA analysis, in forensic investigation, especially in cases like sexual assault, identifying body fluids plays a crucial role in uncovering the truth. So firstly, under DNA analysis, forensic scientists isolate DNA from fluid such as blood, semen or saliva. This allows them to match the DNA to a suspect or victim, providing strong objective evidence that can link individual to the crime scene or eliminate them from suspicion. Secondly is body fluid identification contribute to building accurate timelines. By analyzing how much DNA has degraded or by detecting time-sensitive biological markers, investigators can estimate when the fluid were deposited. This helps place events in chronological order and support the reconstruction of what happened. And lastly, body fluid evidence is vital in corroborating testimony. It can confirm or contradict that the account given by the victim or suspect help to validate statement or uncover inconsistencies. So this is often critical during both the investigation and in court proceeding. So in short, the identification biological fluid is not just about detection, but it's a cornerstone of forensic science that strengthens the reliability of DNA analysis, which establish timing and support the search for the truth.
So now, let's continue to the biological fluid overview. In sexual assault cases, identifying biological fluid is crucial. First is saliva. Saliva may be present from acts like kissing or biting. It contains the enzyme called enzyme amylase, which helps in detection and can be found on victim's skin or even clothes. Second is semen. Semen is one of the most important fluids in forensic sexual assault investigation because it carries both sperm cells and proteins from prostate that can make it rich in forensic markers. Last but not least is vaginal fluid. Vaginal fluid is a mucosal secretion that often left behind during intercourse. Though it is harder to detect than semen, it contains unique epithelial cells and bacterial DNA that can help confirm sexual contact.
Next is about forensic workflow using biological samples. The first step is visual examination and sample collection. We look for any visible stains, injuries or signs of biological fluid using the naked eye or alternative light source, such as UV light. Then we collect the samples using stereo swabs, gloves or proper personal protective equipment. The samples must have proper labeling in order to maintain the chain of custody. The second step is doing the presumptive assay, which is the quick screening to suggest the presence of a specific body fluid. The third step is doing the confirmatory assay to confirm the identity of the body fluid. The fourth step is blood typing. Various blood typing including ABO blood grouping was used for person identification when DNA typing was not available. But nowadays, this type often being excluded but still being helpful for narrowing down suspects for DNA typing because it can be determined from bloodstains, saliva, semen and also hair. The last step is DNA typing that based on the short tandem repeats and sex determination on the amylogenin gene, and there is no doubt about their personal identification power.
One of the most used tests is the feather-based test, which reacts to the enzyme called enzyme amylase. This enzyme is highly concentrated in saliva and causes a color change when detected. Similarly, the starch iodine test shows a color shift when amylase breaks down starch, providing a quick way to screen for saliva. However, both are presumptive tests, meaning they are not specific enough to confirm saliva on their own. For more reliable results, forensic scientists use RNA profiling. These methods detect genes such as staterin or STATH and histamine 3 or HTN3 genes which are only expressed in salivary glands. An even newer and more sensitive approach is miRNA analysis, which use small, stable RNA fragments that are highly specific to saliva and also can survive environmental degradation. Another advanced tool is DNA methylation profiling. This examines the unique epigenetic patterns found in saliva cells, making it possible to distinguish saliva from other fluids with high accuracy.
Now, semen identification. Semen is the most commonly examined body fluid in sexual assault cases. Fresh semen has a distinct odor, while dry stain appear pale yellow or gray and maybe stiff or shiny. When the location or quantity of semen is unclear, forensic investigators often use a variable wavelength light source like a polylight to help locate potential stains. The examination begins with a presumptive test for acid phosphatase, an enzyme found in high concentration in seminal fluid. This is typically done using colorimetric reaction, such as those involving alpha-naphthyl phosphate with fast blue B, which produce a visible color change if acid phosphatase is present. If this test is positive, a confirmatory test, such as microscopic analysis of the staining to identify sperm cells. Several staining techniques are used to highlight sperm under a microscope, including Beigey, Cornstalkis, Christmas tree stain, and methicillin and eosin. These methods help confirm the presence of semen and support forensic evidence in sexual crime investigations. Vaginal fluid identification can be more challenging compared to other body fluids because it doesn't have unique proteins that clearly show its presence. This makes more harder for a forensic expert to confirm if the vaginal fluid is part of the evidence. To detect it, scientists often look at DNA of bacteria that are commonly found in the vagina. So this includes Lactobacillus crepitus, Lactobacillus jacini, and Atypobium vaginae. These microbes can be identified using PCR or DNA sequencing. Another method involves looking at molecular biomarkers like microRNA and DNA methylation patterns. So this can help indicate the presence of vaginal fluid more accurately.
In another interesting case, a sexual assault investigation that had gone cold for more than 15 years was finally reopened and solved thanks to the advancement in molecular biology. So on that time, the original investigation, the biological evidence was too degraded and couldn't be used effectively. But years later, scientists used mRNA based on body fluid identification technique to re-examine the evidence. They were able to detect the vaginal fluid and semen in mixed and degraded sample using specific gene expression markers unique to each fluid. So this allowed investigators to link the DNA back to the suspect, ultimately bringing justice to the victim after so many years. So this case highlights how new technology like mRNA profiling can give a new life to old cases, especially when traditional method fails due to the poor sample quality.
Next is regarding the challenges that are often arise when analyzing biological evidence. Firstly, the biological evidence is often found in degraded conditions where environmental exposure such as heat, moisture, sunlight, or microbial activity can cause DNA and RNA to degrade. This reduces the effectiveness and quality of both body fluid identification and DNA profiling. Biological evidence is often deposited on various substrates such as forest, which is clothing or bedding, or non-forest, which is glass and metals. So the substrate type affects absorption, preservation, and recovery efficiency of the biological material. Biological fluids might also present as mixtures of two or more body fluids, making the detecting and distinguishing between body fluid require highly specific markers such as tissue-specific mRNA, miRNA, or proteomics. The last challenge is that biological fluids may not be visible to the naked eye. The use of alternative light sources is needed to induce fluorescence in the certain body fluids. However, too much exposure to such light source can cause damage to the DNA in the stain.
New forensic methods are being developed to protect evidence while testing it. Techniques like FTIR and Raman spectroscopy can identify body fluids without damaging the sample. Scientists are also working to make these tests more stable and reliable, even when the stains are old, dry, or mixed. To use these new methods in court, they must be tested and approved through proper legal and specific standards. In conclusion, identifying body fluids like semen, saliva, and vaginal fluid is vital in forensic work because it connects DNA to specific actions in a crime. Traditionally, tests like enzyme assays and stain are quick and easy, but modern DNA typing works even on tiny or old sample, so body fluid tests must be keep up. That's why experts are now using fast immunotest kit and molecular techniques such as RT-PCR, microRNA, DNA methylation, and bacterial DNA analysis. These new methods are more precise and match the sensitivity of DNA tests, but there still need more research and validation before they are ready for regular use.